Hello guys, today our topic is intracellular accumulation. Intracellular accumulation means that what are those substances that are accumulate inside the cell during the cell injury. For example, if cell get injured, what will be those substances that accumulate? These substances are lipids, proteins and glycogen. Now we will discuss the causes of intracellular accumulation. These are toxin and diabetes, and we will discuss diabetes later. And for MCQ's point of view, remember that diabetes associated with obesity is the most common cause of fatty change in liver. It means that those people that are obese and have diabetes, they have fat accumulation inside the liver. First, we will discuss lipid accumulation. In lipid, two things are accumulated. Number first, triacyl glycerol, and number second, cholesterol and cholesterol esters. First, we will discuss triacyl glycerol accumulation. It is also called steatosis, which is referred to accumulation of triacyl glycerol. It is mostly seen in liver and also seen in skeletal muscles, cardiac muscles, and kidney. How triacylglycerol are accumulated in diabetes? We had already discussed that the cause of intracellular accumulation is diabetes. How triacylglycerol are accumulated in diabetic patients? We know in diabetes mellitus insulin level goes down, and we also know from biochemistry that insulin is the main regulator of lipid metabolism. So if a level of insulin is increases, there will be greater lipid metabolism. If the level of insulin is decreased, there will be low lipid metabolism. Best example is type 1 hyperlipoproteinemia. In type 1 hyperlipoproteinemia, VLDL level increases. And the normal function of VLDL is that it carries triacylglycerol from arteries to organs. So, if level of VLDL increases, there will be more carrier for triacylglycerol. So, triacylglycerol accumulation will occur in the organs. Now we will discuss cholesterol and cholesterol esters accumulation. It is mainly accumulate due to high intake or low catabolism of cholesterol. It is mainly seen in arteriosclerosis and diabetes. In arteriosclerosis, VLDL gene mutation occur. And VLDL have two functions. Number first, we had already discussed that it carries triacylglycerol from artery walls into the organs. And number second, it takes cholesterol from organs into the artery wall. So when VLDL gene mutation occur, cholesterol start accumulating in the artery wall in the arteriosclerosis. Number second, diabetes. We know in diabetes insulin level decreases, and insulin is the stimulator of cholesterol metabolism. So if insulin level decreases, cholesterol metabolism will be decreases. When there is no cholesterol metabolism, cholesterol will start accumulation. Now remember that we know that HDL is called cholesterol. Why it is called good cholesterol? Because it take cholesterol from artery wall into the liver, and VLDL take cholesterol from the organ to the artery wall. Now we will discuss protein accumulation. Uh, we know that normally protein are not filtered in the glomerulus. Only small amount of albumin protein are filtered, and it is reabsorbed back by the cell of proximal convoluted tubules by the process of pinocytosis. Now, protein will accumulate in those diseases in which heavy leakage of protein occur from the glomerulus. The best example is nephrotic syndrome. In this syndrome, heavy leakage of protein from glomerulus occur. If high protein leakage occur, high will be the reabsorption, and protein will start accumulation in the proximal convoluted tubule. For MCQ's point of view, remember that histological appearance of accumulated protein is pink hyaline cytoplasmic droplets. So now, what is hyaline? Hyaline is a cartilage, and it is of two types. It is intracellular hyaline and extracellular hyaline. First, we will discuss intracellular hyaline. For MCQ's point of view, remember that most common example is Mallory hyaline. Most common example of intracellular hyaline is Mallory hyaline, and Mallory hyaline is mostly seen in alcoholic liver diseases. Number second, Mallory hyaline is composed of intermediate filament 8 and 18. Number second is extracellular hyaline. It means that it is accumulate extracellularly. And number second, the best example that it is mainly seen in adenine cystic carcinoma. Now we will discuss glycogen accumulation. Glycogen mainly deposit in glycogen storage diseases, and patient with diabetic nephropathy can get a lesion called Armani Epstein lesion. In this lesion, glycogen are accumulated in the proximal convoluted tubule. Histopathology of glycogen accumulation. Remember that glycogen appear as a clear vacuole in cytoplasm. And number second, the special stain that we use for the glycogen is positive stain. And on this stain, glycogen look pink. The best example of glycogen storage diseases is glycogen storage disease type 1. First, we should know about glycogen storage disease. Glycogen storage disease are group of disease in which those enzymes have defects which are involved in the glycogen metabolism or glycogenolysis. In glycogen storage disease type 1, same glucose 6 phosphatase is absent in the cytoplasmic reticulum. And the normal function of glucose 6 phosphatase is that it converts glucose 6 phosphate into glucose. So, if glucose 6 phosphatase is absent, conversion of glucose 6 phosphate into glucose will not occur, and glycogen will start accumulating because glucose conversion of glucose 6 phosphate into glucose is the last step of glycogenolysis. What are those sites where the glycogen can accumulate? It is cardiomyocytes, beta islets of Langerhans, and renal tubule basement membrane. So if you are new to our channel, please like, share and subscribe.